Hi, today we are going to finish the first trip through the digestive system. In this first trip, we're going to go through the anatomy, and then with our next lecture, we'll pick up on the special topics of movement within the GI tract, followed by secretions of the GI tract, and then digestion and absorption of nutrients. We already started this anatomy trip, so I'll quickly review the first structures and then move on to later structures. Our anatomy trip starts with the oral cavity, along with the teeth. When food is ingested, it then is broken apart by mechanical actions of the teeth and formed into a bolus by the tongue, where it continues down the oropharynx to the pharynx. When food is ingested, it gets moved by the tongue, and the tongue will push that food bolus up against the hard palate, which is made up of palatine bones, as well as the palatine processes of the right and left maxilla. The soft palate is going to, when the swallowing reflex is initiated, the soft palate moves upward and closes off that nasal cavity. So food is going to continue through the oropharynx down past laryngopharynx into the esophagus. The tongue is responsible for gripping and repositioning food during chewing. It will also mix the food with saliva and form a bolus, and then swallowing is initiated. It is also responsible for speech and taste. There are two sets of skeletal muscles, intrinsic, which are not attached to bone. These are going to be important in speech, especially the formation of consonants, as well as initiating the swallowing reflex. And there are also extrinsic muscles of the tongue, which will allow us to protrude or stick out our tongue, retract it, and move it side to side. There are 4 sets of papillae, and the most numerous are the filiform papillae, which are all over the tongue and are responsible for friction. The other 3 sets, fungiform, vellate, and foliate, are going to house the taste buds. We'll talk more about the composition of saliva when we get to secretions, but saliva does begin the digestion process by the secretion of amylase. It also helps to moisten food and allows the food to be compacted into a bolus. Saliva will also dissolve food chemicals for taste. Most of the saliva is produced by the 3 major extrinsic salivary glands, the parotid, submandibular, and sublingual. There are some other buccal glands that have a smaller contribution. There are 2 types of secretory cells. Serous cells produce watery secretions and a bit of mucin, which we'll talk about a little bit later, as well as mucus cells, which produce mucus. The parotid and submandibular glands contain mostly serous glands, but the sublingual gland contains mostly mucus cells. We talked about deciduous teeth. These are our primary teeth that fall out. There are 20 of them, and then those are going to be followed by permanent teeth, and there are 32 of those. The pharynx is composed of 3 different sections. The nasopharynx is not considered part of the digestive system. So the oropharynx is very important because that is where food is going to start its pathway down the alimentary canal. And then we have the laryngopharynx, which gets closed off by this epiglottis here when we swallow. The pharynx is made up of stratified squamous epithelium, mucus glands, and skeletal muscle. The laryngopharynx becomes the esophagus, a muscular tube leading to the stomach. and it's going to join the stomach as the lower esophageal sphincter at the cardia portion of the stomach. The esophagus has a stratified squamous epithelium to protect it from abrasion, but then it also is interesting in that its muscular layer changes from being comprised of skeletal muscle for the superior third to smooth muscle for the inferior 2/3. As we would expect, glands secrete mucus to help aid the bolus in sliding along the esophagus. Once the food bolus reaches the lower esophageal sphincter, which is about here, it will then continue into the cardia portion of the stomach. The fundus is the part that is located above the juncture of the esophagus, so it's kind of a balloon up here. Then we have the body of the stomach leading to the pyloric section ending in the pyloric sphincter. The stomach is made up of these rugae, these folds, which allow, the stomach actually becomes quite small when it's empty. It will only contain about 50 mils of fluid, but then when food enters the stomach, it can go up to about 1 liter. So it has a great capacity for distension, and that's because of these rugae. There is an additional layer of muscle in the stomach. So the rest, so far, what we have seen in like the esophagus and what we'll see in the small intestine will be a longitudinal layer, and that is interacting with a circular layer. However, the stomach has a third layer. This is called an oblique layer. It's fairly thin, and in some places it can be incomplete. But this third oblique layer is going to aid in the churning action of the stomach. There's a lesser curvature and a greater curvature. And the design of the stomach, we'll talk a little bit more about this when we get to motility, is the stomach contracts in such a way that there is just a little bit of churning up here in the fundus and body, but then a much more extensive churning in this lower section, in the pyloric section. Food is going to be secreted at a relatively consistent rate to enter the, you can either say duodenum or duodenum. With the stomach, there are, as I mentioned, there are 3 layers of smooth muscle. The additional layer is that oblique layer of smooth muscle. The stomach also contains gastric pits and gastric glands. The gastric pit is the part of this structure that is closest to the stomach surface. It's where the contents from the gastric glands are going to collect and be secreted. So the gastric glands are going to contain mucus cells, parietal cells, and chief cells, along with the enteroendocrine cells. So the gastric gland contains multiple cell types that will produce multiple secretions. Then all of those good secretions are going to gather into this gastric pit and be secreted. The other thing that happens in this gastric pit is these products from all of these glands. Many of them are secreted in peptides that need to be clipped to become activated. And some of them, that kind of happens with in the acidic environment in these pits. So we don't want to secrete active pepsin because pepsin is going to digest proteins. That we don't want that, because that would digest the cells that are producing it and the ones that are adjacent. So instead, we want to secrete precursor products that then get activated in this gastric pit so that we're not digesting our stomach. The 4 gastric glands that you need to know are going to be the parietal cells. And the parietal cells are going to be, they're going to be more numerous towards the top. So as we get closer to the gastric pit, and that's because they secrete hydrochloric acid. So you don't want them here at the bottom, you want them here at the top. The hydrochloric acid is going to activate pepsinogen and create pepsin, which will denature proteins and start in protein digestion. HCl will also kill bacteria, and the parietal cells will produce intrinsic factor, which is required for vitamin B12 absorption. The chief cells are going to produce pepsinogen, so this pepsin precursor, and that's going to be at the, more at the end of the gastric glands. Mucus neck cells are going to produce mucus, and they are located here. This is called the neck of the gland. So we have the chief cells secreting pepsinogen. We have parietal cells secreting hydrochloric acid, and then we have mucus cells secreting mucus. So as the pepsinogen travels up through this gastric gland, it will get activated as it passes into this acidic environment. And then by the time it hits the mucus neck cell, we have a highly acidic environment that also contains protein digesting enzymes. The enteroendocrine cells are also located here at the bottom, and they secrete a kind of a wide array of hormones, gastrin and serotonin, and a few other hormones that we're not going to go into. Okay, so the main ones to keep in mind, because we're really focusing on digestion right now, are the parietal cells, the chief cells, the mucus neck cells. And this mucus is slightly acidic, by the way, but to know what they produce, so hydrochloric acid, pepsinogen, and an acidic mucus, to know where their location is, that at this kind of at the bend, at the deep section, this deep U section, is where you'll find the chief cells. Slightly further up that gastric gland, you'll find the parietal cells. And then the closest to the gastric pit will be the mucus cells. So you do need to know what they produce, as well as where you would find them, because that order is very important in getting a functioning pepsin enzyme, but without digesting the stomach glands and muscle layer. There is a mucosal barrier that is produced. It is a double layer of mucus, and it contains bicarbonate in between those mucus layers. These are, this helps to protect the stomach from getting digested. There are also tight junctions of the epithelium to prevent digestive enzymes from going anywhere other than the stomach lumen. And then there's a rapid replacement of the stomach epithelial lining. The stomach epithelium is replaced every 3 to 6 days. So those are some of the protective mechanisms of the stomach. Vomiting is called emesis, and it is a reflex. There can be multiple stimuli. It can be either due to over distension of the stomach, so someone actually physically eats too much, a bacterial infection, a viral infection, sometimes spicy foods, and then alcohol or drugs your body will try and rid itself of. The diaphragm and abdominal muscles are going to contract. This cardiac sphincter relaxes, and the uvula will close off the nasal cavity. Repetitive vomiting can lead to alkalosis due to a loss of HCl. Moving on to the small anatomy. So we've had food, it's become a bolus in the mouth, and it has traveled down the esophagus, through that lower esophageal sphincter, through the stomach, to the pyloric sphincter, and it is now going to enter the small intestine. The small intestine begins at the pyloric sphincter and ends at the ileocecal valve. So the end of the stomach to the beginning of the large intestine. There are 3 divisions, the first being the smooth muscular tube section, that is the duodenum. Then we have about 8 feet of jejunum, ending in 12 feet of ileum. And that ileum joins to the large intestine at the ileocecal valve. So you do need to know the beginning valve, the ending valve, and the order of the 3 sections. The small intestine and the large intestine are both heavily modified for their functions. For the small intestine, those modifications tend to occur more on the microscopic level. So there are a lot of modifications made to the small intestinal epithelial cells and kind of their arrangement. These structural modifications will increase surface area to increase nutrient absorption. The three of the structural modifications are called the plicae circularis, villi, and microvilli. The plicae circularis these are deep circular folds, they're permanent folds of the mucosa and the submucosa. And so they don't distend with the food and become flattened like the rugae do. These are going to be permanent folds. It's due to the arrangement of the mucosa and the submucosa in the small intestine. And this is going to create a slowing of food movement so that there is an increase in absorption time. The villi are finger-like extensions of the mucosal layer. So this is going to be the arrangement of the mucosa. From these villi are microvilli, and microvilli are created by actin fibers that form together, or they link together underneath the plasma membrane to make this brush fuzzy border that increases surface area. So villi is the arrangement of the mucosal layer. Microvilli are due to an underlying actin web of the plasma membranes that provide tension on them to increase the surface area. So you should be familiar with these 3 modifications and what they look like and what their function is. So the plicae circularis are permanent folds because of the arrangement of the mucosa and submucosa. Its role is to slow the movement of chyme and allow for absorption of nutrients. A villus or villi are the arrangement of the mucosa. And so they arrange in these finger-like projections. And each villus is going to have capillaries and a lacteal, which we'll talk about when we get to absorption. We'll also talk about intestinal crypts when we get to absorption. Then each one of these, so there's hundreds of simple columnar epithelial cells that are lining each villus. This is a singular simple columnar epithelial cell of the small intestine, and then its plasma membrane has been made into microvilli. So a villus is made up of hundreds upon hundreds of simple columnar epithelial cells to create this structure. Microvilli are extensions of the plasma membrane that are on a singular simple columnar epithelial cell. We're going to diverge off of our pathway just a little bit to discuss the anatomy, the arrangement of the gallbladder, the pancreas, and the duodenum. To chemically digest fats, beginning in the duodenum, we need bile salts from the gallbladder, which are really from the liver, as well as pancreatic lipase from the pancreas. So we are getting a little ahead of ourselves as far as digestion, but to digest fats, we need to break them apart using lipase, and then we need to emulsify them using bile salts. So in order to digest fats, we're going to need both bile salts as well as lipase. The gallbladder is kind of on this posterior aspect of the liver, and then the pancreas is going to slide just underneath the stomach. The gallbladder stores bile. The liver makes bile. So the gallbladder is this thin-walled green muscular sac on the ventral surface of the liver, and it is going to store and concentrate bile. It releases bile via the cystic duct, which flows into the common bile duct. We're going to go over that duct system. So bile is made by hepatocytes. Hepatocytes are the functional cells of the liver. The liver is going to be kind of up here. It has 4 lobes, a right lobe, a left lobe, a caudate lobe, and a quadrate lobe. And these hexagonal hepatocytes, sorry, I know my drawing's not great, are going to produce bile. That bile then goes through canaliculi, and it is going to get collected into ducts, which eventually are going to flow into the right and left hepatic ducts of the liver. The right and left hepatic ducts are going to form a common hepatic duct. The bile flows from those right and left hepatic ducts through the common hepatic duct and then up into the cystic duct where it is stored in the gallbladder. When we have a fatty meal and we need bile to emulsify. That fat, the gallbladder then releases bile back through the cystic duct to the common hepatic duct. You might have noticed that the cystic duct is bidirectional. It is going to allow for bile to flow into the gallbladder and back out through the gallbladder as needed. There can also be some flow from the common hepatic duct into the common bile duct. But in general, most of the bile salts are going to be stored in the gallbladder and secreted through the cystic duct to the common bile duct. The common bile duct fuses with the pancreatic duct right here at the hepatopancreatic ampulla. There's also this accessory pancreatic duct that has a very minor role in adding enzymes into the duodenum. You do need to know the order of ducts, as well as if you are given a diagram, what duct could be labeled. So this is just another blow up of that same diagram. The liver is going to produce bile. It flows through bile canaliculi. It will empty into either the right or left hepatic duct, which then come together to form this common hepatic duct. The bile then goes back up the cystic duct to get stored in the gallbladder. If we have a fatty meal, the gallbladder gets stimulated to release bile back through that cystic duct into the common hepatic duct, where it joins with the pancreatic duct at the hepatopancreatic ampulla. The duodenum is about 10 inches long, and once it makes this curve around the pancreas, it then is going to turn into the jejunum. The pancreas lies deep to the greater curvature of the stomach. Its head is encircled by the duodenum and its tail touches the spleen. So it is here. The pancreas has endocrine functions that we've talked about in 1141 with the production and release of insulin and glucagon. For the digestive system, we will be focusing on its exocrine functions. It secretes pancreatic juice, which is made up of several enzymes, which we'll be discussing in the secretion trip. The pancreas is made up of these acinar cells, which are going to produce enzymes in these using these zymogen granules. And we'll get into the specifics of the enzymes when we talk about secretions. The small intestine, we went from the duodenum to the jejunum to the ileum, and then the ileum ends at the ileocecal sphincter. The ileocecal sphincter is going to protect the small intestine from the bacteria of the large intestine. So there's quite a colony of bacteria living in the large intestine. And a reservoir of that bacteria is maintained in the appendix, so that if bacteria gets wiped out by, let's say, antibiotics, there is a reserve that is not as affected there. Maybe it gets wiped out by something that's a harmful bacteria, but the appendix has a reserve of bacteria that can be used for recolonization. The cecum is the first pouch-like part that the ileum dumps into, and then it progresses along the ascending colon. Then we have I'll show you the flexures to transverse to descending to sigmoid to the rectum, ending in the anal canal. The ileocecal valve is right here. Okay. So we have the ileum that is going to then get closed off by this ileocecal valve. The food material, which the main function of the large intestine is to store food until it can be excreted. There will be a little bit of reabsorption of water, but not much. So food is going to enter at the cecum, travel up through this ascending colon. Then there is the right colic, also known as the hepatic flexure, leading to the transverse colon. Then we have the left colic flexure, also known as the splenic flexure, leading to the descending colon. Then the sigmoid colon is this section here to the rectum, and the rectum has 3 valves in it, and then leading to the anal canal. As I mentioned, there are distinct regions: the ascending colon to the hepatic flexure, to the transcending colon, to the splenic flexure, to the descending colon, to the sigmoid colon, ending in the rectum and then the anus. The transverse and sigmoid portions are anchored via mesenteries called mesocolons. We'll talk about the rectum and the anal canal when we get to defecation. The large intestine also has 3 unique features. This looks like rope-like structure here is the tenai coli, and this is what is left of the longitudinal muscle from that muscular layer. We have circular and we have longitudinal, and these are the remnants of the longitudinal muscle fibers. The tone of this tenai coli is going to create pouches. And these pouches are called haustra. So a singular is haustrum, several of them are haustrum. The third unique feature are these epiploic appendages. These are just fatty pieces that hang off of the tenai coli, and no one really knows what their purpose is. But these are the 3 unique features: the tenai coli, which are 3 bands of longitudinal smooth muscle. The tone of the tenai coli create pocket-like sacs called haustra. And then hanging off of the tenai coli are epiploic appendages, which are fat-filled pouches with no known function. The colon mucosa is a simple columnar epithelium, except in the anal canal, it'll go to a stratified squamous for protection. There are numerous crypts lined with mucus-producing goblet cells, and that keeps the waste moving along. The anal canal is a stratified squamous epithelium, as I mentioned, for it's a protective surface. Anal sinuses also excrete mucus and will compress feces. There are these superficial venous plexuses are often get inflamed, and these cause hemorrhoids. Okay, so now we are moving on. We've almost exited the alimentary canal. The rectum has 3 sphincters, and then the anal canal has 2 sphincters. It has an external and an internal. As I mentioned, there are bacterial flora that live in the large intestine. These are bacteria that survive the small intestine, or they can enter the anus, or we seem to accumulate them kind of through especially our younger years. As I mentioned, they get stored in the appendix. Their purpose is to colonize the colon, preventing pathogenic bacteria from also colonizing the colon. They aid in fermenting indigestible carbohydrates. They do produce gas in their metabolism, and then they synthesize B complex vitamins and vitamin K, which is necessary for the liver as well as blood clotting. Functions of the large intestine: other than digestion of bacteria, no further digestion takes place. Vitamins, water, and electrolytes are reclaimed. Its major function is propulsion of fecal material. Though essential for comfort, the colon is not essential for life. Waste can be collected in something called an ostomy bag. And there is about 12 to 14 hours of travel time. Defecation is the initial stimulus, is distension of rectal walls by feces. And then this is going to create a reflex pathway. So the first event is that, so we had our ascending colon, our transverse, our descending to our sigmoid. And then now there are actually 3 sections to the rectum. And as the walls start to stretch due to fecal contents, then an afferent nerve is activated. That sensory nerve is going to send information up to the cortex and say, hey, is this a good time to go to the bathroom? And then that cortex will send a message down, yes or no. But the sensory nerve fiber is going to also have a reflex pathway within the spinal cord, leading to involuntary relaxation of the internal sphincter. The external sphincter is under voluntary motor control. And so when this pathway also sends notification up to the brain will then send a signal down and say, okay, it's a good time to go to the bathroom, or no, we need to keep that sphincter closed. The internal anal sphincter, as one would expect, is made up of smooth muscle because it is under involuntary nervous control. The external sphincter is made up of skeletal muscle, as you would expect, because it is under voluntary motor control. So you do need to know which sphincter is smooth muscle and controlled by the parasympathetic division, and which sphincter is made up of skeletal muscle and controlled by the voluntary nervous system. That is where we are going to stop for today, and we will pick up with motility and move on to secretions and maybe get to absorption at our next lecture.